Today we're going to unravel the labyrinth between two key signaling pathways through a novel complex my laboratory recently discovered. Signaling pathways throughout the human body are much like the complicated pathways of train systems, with starting points, connections, longer routes, and loops. Unlike these maps, there are hundreds of signaling pathways throughout the human body, many of which are barely understood. The calcium and G protein signaling pathways are two of the most expansive pathways in the human body. Both are essential to almost all processes in our cells and bodies to maintain proper health. Both signaling pathways in total are linked to more than 50 human diseases, syndromes, and cancers if not working properly. Delving first into calcium signaling, when the intracellular concentration of calcium rises, the calcium-binding protein calmodulin is activated by 4-bound calcium ions. It then continues on to bind hundreds of cellular proteins to regulate their functions. Calmodulin is considered a master regulator of cellular processes and is of high evolutionary importance. It even controlled the beak shape of Darwin's finches. G-protein signaling is initiated by G-protein-coupled receptors, GPCRs for short, and there are around 850 different ones in our cells. GPCRs consist of 7 transmembrane helices with portions inside and outside the cell. They have an intracellular G-protein attached consisting of alpha, beta, and gamma subunits. The G-alpha subunit controls the duration of G-protein signaling and is known to be involved in calcium signaling, but not with calmodulin. As there is such a wide range of GPCRs that play a crucial role in health and disease, around 700 approved drugs on the market target them. This is 30% of all drugs. When inactive, the GPCR plus G-protein is an intact unit with G-alpha bound to GDP. When an extracellular ligand binds, the GPCR is activated, exchanging GTP for GDP on the alpha subunit. When bound to GTP, the alpha subunit separates from the entire unit, which initiates downstream G-alpha and G-beta gamma signaling, also known as G-protein signaling. The intrinsic GTPase activity of G-alpha, or cutting off one phosphate group, which converts GTP back to GDP, can be accelerated by certain proteins terminating the signaling pathways. Any protein that affects the speed of GTP hydrolysis on G-alpha affects the entire G-protein signaling pathway. All of my research points to one conclusion. Calmodulin binds to the G-alpha subunit of G-proteins. My research has 2 separate goals, the first of which is to characterize the structural interaction through analytical methods. The second of which is to define the functional consequences. Does it shorten signal duration? Are there farther downstream effects on signaling? Does it affect the activation or inactivation of G-proteins? What we now know is that our newly discovered interaction between calmodulin and G-alpha has brought about a novel connection between the calcium and G-protein signaling pathways. This interaction has implications towards multiple complicated diseases such as Parkinson's, could lead to the development of therapeutics in currently limited fields of treatment, and lastly, understanding this novel interaction will lead to more sophisticated models to aid in the next generation of effective pharmaceuticals. To conclude, thank you so much to the ARCS Foundation for your consideration.